There is a world beneath the world, not a metaphor, not a philosophical idea, a literal, physical world, vast, ancient, largely unexplored, lying beneath the surface of the ocean, in depths so great that sunlight has never reached them, in pressures so extreme that unprotected human bodies could not survive them, in temperatures so cold that life, by any ordinary reckoning, should not be there at all. And yet life is there, not sparse, struggling, barely holding on life, abundant life, diverse life. Communities of extraordinary creatures, some familiar in their general shapes, some unlike. Anything that lives in the sunlit world above, gathered around structures that rise from the seafloor like the towers of a drowned cathedral, sustained by chemistry rather than sunlight, ancient beyond easy comprehension. Tonight, we are going to descend into that world together, slowly, gently, the way the deep ocean itself descends, not in a sharp plunge, but in a long, gradual darkening, a progressive quieting, a deepening of pressure and cold and mystery that takes hours to complete. We will not rush. There is nothing to rush toward down here. Time moves differently in the deep ocean. Processes that take seconds in the sunlit surface world take years, decades, centuries in the abyss. The creatures that live down here are, many of them, extraordinarily long-lived. Some of the corals that grow on seamounts and ridges in the deep ocean are thousands of years old, older than most human civilizations. Some of the individual clams living around hydrothermal vents may live for centuries. The deep ocean is a place that does not hurry. It suits us well tonight. If you are listening to this as you prepare for sleep, then you are already beginning the descent. Your body is slowing, your breathing is deepening. The activity of the day is receding above you, like the sunlit surface of the ocean, receding as you sink beneath it. Below, ahead of you, is the quiet, the dark, the deep. Let us go there together. The ocean covers approximately 71% of Earth's surface. It has an average depth of about 3,700 meters, nearly 4 kilometers. Its deepest point, in the Mariana Trench in the western Pacific Ocean, reaches nearly 11 kilometers below the surface. At that depth, the pressure is more than 1,000 times the pressure at sea level. Most of this ocean is deep. More than 90% of the oceans, its volume lies below 200 meters, below the depth at which sunlight can meaningfully penetrate. This vast, dark, cold, high-pressure world is called the deep ocean, and it is, by any measure, the largest living space on Earth. It is also the least explored. More of the deep ocean floor has been mapped in less detail than the surface of Mars. We know more about the topography of the Moon than we do about the topography of the seafloor in many parts of the world's oceans. What we have seen, in the places we have visited, is extraordinary. We have found mountain ranges longer than any on land. We have found plains that stretch for thousands of kilometers without interruption, covered in a fine sediment that has been settling, particle by particle, for millions of years. We have found trenches that plunge to depths that would bury Everest with room to spare. And we have found, rising from the seafloor in specific locations, the structures that are the heart of tonight's journey, the hydrothermal vents. These are the towers we mentioned, the structures that rise from the seafloor where superheated water, enriched with minerals from deep within the earth, rises through cracks in the ocean floor and meets the cold of the deep, where they build, over years and decades, chimneys of mineral deposits that can reach tens of meters into the water above, and where, around their bases and along their sides, communities of life, unlike anything in the sunlit world, gather in extraordinary density. We call these communities vent ecosystems, and they are, in the truest sense, the ancient forests of the deep ocean floor. Not forests of trees, of course, but communities as complex, as layered, as interdependent, and as ancient as any forest on the surface. Communities with their own food chains, their own ecological dynamics, their own caste of species found nowhere else on Earth. Communities sustained not by the sun, but by the chemistry of the Earth's interior, by the heat of the planet itself. They were discovered in 1977, and their discovery changed the way science thinks about life, about where life can exist, about what life requires, and about the possibility of life on other worlds. We will spend a long, unhurried time with them tonight. But first, let us descend, gently, through the layers of the ocean, from the sunlit surface to the lightless deep. Imagine you are at the surface of the Pacific Ocean, somewhere in the middle of that vast blue expanse, far from any coast. The water around you is warm and bright. Sunlight penetrates to perhaps 100 meters, shimmering, shifting, turning the water column into a cathedral of blue light. Schools of fish move in the middle distance. Seabirds work the surface above. The world up here is busy, luminous, alive in a way that is immediately legible to human senses. You begin to descend. At 50 meters, the light has shifted from white to blue. The warmth of the surface is fading. The sounds of the surface world, wind, waves, the distant engines of ships, are muffled, diminished. Below you, the blue deepens into something darker. At 200 meters, you cross a threshold that oceanographers call the mesopelagic zone, the twilight zone of the ocean. Light still reaches here, but only barely. It is a dim, blue-violet twilight, not enough to support photosynthesis, barely enough for eyes adapted to the deep to navigate by. The temperature has dropped significantly. The pressure is already more than 20 times the pressure at the surface. Strange creatures live in this twilight zone. Fish with enormous eyes, designed to catch every last photon of the fading light. Creatures that produce their own light, bioluminescence, in cold chemical fires that flicker in the dark. Jellyfish trailing long, translucent tentacles, siphonophores, colonial organisms that can reach lengths of 40 meters or more, the longest known animals on Earth, drifting in the dim like ghostly aerial photographs. At 1,000 meters, the last light is gone. This is the bathypelagic zone, the midnight zone. Here, no sunlight has ever reached. The darkness is absolute in the way that very few places on Earth are absolutely dark. The temperature is a few degrees above freezing. The pressure is 100 times the surface pressure, and yet life continues, adapted to the dark, to the cold, to the pressure. Fish with hinged jaws that can swallow prey larger than themselves. Anglerfish with their bioluminescent lures dangling before their toothy mouths. Vampire squid, not a true squid, but a unique cephalopod unlike any other, drifting through the dark with its dark cape spread wide. The bathypelagic zone extends to about 4,000 meters. Below that begins the abyssopelagic zone, the abyss, still colder, still darker if that is possible, still higher in pressure. The seafloor, at these depths, is a vast plain of fine sediment, the accumulated rain of organic material that has been falling from the sunlit surface world above for millions of years. Marine snow, scientists call it, the constant slow precipitation of particles, dead organisms, fecal matter, shed scales and shells and the remains of countless surface world lives, drifting slowly downward through kilometers of water, taking weeks to reach the floor. This marine snow is the primary food source for most of the deep ocean floor. The creatures that live on the abyssal plain, sea cucumbers, brittle stars, strange worms, pale crabs, feed on this fallen material, living on the afterglow of sunlight that never directly reach them. They are the consumers of the deep, sustained by the productivity of the distant surface, processed through kilometers of water column and weeks of slow descent. This is the deep ocean as it exists across most of its vast extent. Cold, dark, quiet, sustained by the downward drift of organic material from above. But there are places where the story is different. Places where the seafloor itself is the source of energy, rather than the surface far above. Places where the Earth's interior reaches upward, providing heat and chemistry to communities that need nothing from the sun. These are the hydrothermal vents, and they are waiting for us, down in the dark, at the edges of the tectonic plates. To understand hydrothermal vents, we need to understand the movement of the seafloor. The Earth's outer layer, the lithosphere, is not a single, continuous shell. It is broken into a series of large and small pieces called tectonic plates. which move very slowly across the surface of the planet, driven by the convection of the hot mantle beneath them. These plates are in constant, imperceptibly slow motion, moving at speeds comparable to the rate at which your fingernails grow. Where plates move apart, at what geologists call spreading centers or mid-ocean ridges, new seafloor is created. Hot mantle material rises from below, fills the gap between the separating plates, and cools to form new oceanic crust. This process of seafloor spreading is happening right now along the mid-ocean ridge system, a continuous chain of underwater mountains that winds through every ocean on Earth, covering a total length of more than 60,000 kilometers. The mid-ocean ridges are the longest mountain ranges on Earth. Most of them lie entirely underwater, unvisited and largely unmapped. They are geologically active, marked by earthquakes and volcanic eruptions as new crust is continuously created, and along their axes, where the seafloor is youngest and the heat from below is most intense, they are dotted with hydrothermal vents. Here is how a hydrothermal vent forms. Cold seawater percolates downward through cracks and fissures in the young oceanic crust, drawn toward the heat below. As it descends, it is heated, sometimes to temperatures exceeding 400 degrees Celsius. At these temperatures, the water dissolves minerals from the surrounding rock, becoming enriched with metals, iron, copper, zinc, manganese, and with chemicals like hydrogen sulfide, the compound that gives rotten eggs their characteristic smell. This superheated, mineral-rich fluid then rises back upward, driven by its own buoyancy, and emerges from the seafloor through openings in the rock. When it meets the cold, 4-degree water of the deep ocean, the dissolved minerals precipitate out, forming the mineral deposits that accumulate around the vent opening, building the chimney structures that can reach heights of tens of meters. The most dramatic of these chimneys are called black smokers. The fluid emerging from them is so hot, sometimes above 400 degrees, and so rich in dark sulfide minerals that it looks exactly like black smoke billowing from a chimney. It is not actually smoke, of course. It is superheated mineral-rich water, instantly precipitating its dissolved load as it meets the cold ocean. But the visual impression is striking. A tower of dark stone rising from the seafloor, belching what looks like dense black smoke into the water above. White smokers are cooler, their fluid temperatures are lower, and their mineral content different, producing white or pale gray precipitates. They tend to be more structurally complex, sometimes forming elaborate, delicate formations, branching towers, fluted columns, fragile mineral lace that would be at home in a science fiction landscape. There are also diffuse flow vents, areas where warmer water seeps gently through the seafloor without forming dramatic chimneys, emerging in scattered, subtle flows across wider areas. These diffuse flows, though less visually dramatic than the black smokers, are actually more important in terms of the total heat and chemical energy they deliver to the deep ocean, and they support some of the densest communities of vent organisms. The vent structures themselves are not permanent. They grow and shift and eventually collapse as the underlying geology changes. Vents can become inactive, the heat below them fading, the mineral-rich fluid no longer rising, and when a vent goes inactive, the community that depended on it must disperse or die. New vents form elsewhere along the ridge, and the process begins again. In this sense, the hydrothermal vent ecosystems are dynamic, appearing, flourishing, declining, and disappearing on timescales of years to centuries, driven by the slow movements of the tectonic plates below. In February 1977, a small submersible named Alvin descended into the Galapagos Rift, a section of mid-ocean spreading center in the eastern Pacific Ocean, roughly 450 kilometers northeast of the Galapagos Islands. The scientists aboard were geologists and geochemists. They were looking for hydrothermal vents. They had detected warm water anomalies in previous surveys that suggested vents might be present. They expected to find hot water emerging from the seafloor, interesting geology, perhaps some unusual mineral formations. What they found instead changed biology forever. The seafloor around the vents was carpeted with life. Life in extraordinary density, in a place where, by all established understanding, life should barely exist. Giant clams, up to 30 centimeters across, clustered around the vents in numbers that seemed impossible. Mussels formed thick beds. White crabs moved among the clusters. And most remarkable of all, dense thickets of tube worms, up to 2 meters long, their bright red plumes extending from white tubes, swaying gently in the vent currents. The scientists aboard Alvin were not biologists. They had no sampling equipment prepared for biological material. They made do with what they had, collecting organisms in bags, preserving them in vodka purchased in the Galapagos before the dive, improvising as best they could in the face of a discovery they had not anticipated. The organisms they brought back were unlike anything previously known, and the question of how they survived, what they were eating, what powered their extraordinary abundance in the absence of sunlight, was immediately and deeply puzzling. The answer, pieced together over the following years by a generation of biologists working on the vent samples, was chemosynthesis. Chemosynthesis is the process by which certain bacteria and archaea, microorganisms, extract energy from chemical reactions rather than from sunlight. Specifically, at hydrothermal vents, these microorganisms oxidize hydrogen sulfide, the chemical released in abundance by the vent fluid, using oxygen dissolved in the surrounding seawater. The energy released by this chemical reaction is used to fix carbon dioxide into organic molecules, just as photosynthesis fixes carbon dioxide using the energy of sunlight. These chemosynthetic microorganisms are the primary producers of the vent ecosystem, the equivalent of plants in a sunlit ecosystem. They capture energy from the chemical environment and store it in biological molecules, making it available to the rest of the food chain. Some of these microorganisms live freely in the water around the vents, forming thick mats on the seafloor, growing in the plumes of vent fluid rising into the water column, colonizing every available surface. Others live inside the bodies of vent animals, in specialized organs, in the cells of gill tissues, in symbiotic relationships of extraordinary intimacy. The tube worms, those remarkable red-plumed creatures that so astonished the Alvin scientists, have no mouth, no gut, no digestive system of any kind. Their entire interior is occupied by a specialized organ called a trophosome, packed densely with chemosynthetic bacteria. The tube worm provides the bacteria with the sulfide and oxygen they need to carry out chemosynthesis, and the bacteria provide the tube worm with the organic compounds it needs to survive. This symbiosis, bacteria living inside animals, powering their hosts with chemical energy, turned out to be a fundamental organizing principle of vent ecology. The giant clams have chemosynthetic bacteria in their gills. The mussels have them too. Many vent organisms carry their food source inside them, a biological solution to the fateful challenge of living in a place where the food gradient runs in the opposite direction from the Drees. Fine challenge world above. The 1977 discovery at the Galapagos Rift was not just the discovery of a new ecosystem, it was the discovery of a new principle: that life does not require sunlight, that the energy of the Earth's interior, expressed through chemistry, can sustain entire communities of living things, independent of any photosynthetic foundation. This principle has implications that reach far beyond the deep ocean floor of Earth. Let us spend a moment with the chemistry itself, the invisible engine that powers everything we are going to encounter tonight. Hydrogen sulfide. The molecular formula is H2S, 2 hydrogen atoms, 1 sulfur atom, a simple molecule, and one that most living things, if exposed to it in high concentrations, would find immediately lethal. It is toxic, in sufficient quantities, to most organisms that evolved in oxygen-rich environments. It is the molecule responsible for the smell of rotten eggs, of volcanic gases, of marshes and sewers, and it is the fuel of the deep. At hydrothermal vents, hydrogen sulfide is produced by the reaction of seawater with hot rock deep in the crust, and it rises in the vent fluid, along with other reduced chemicals, methane, hydrogen, ammonia. When this fluid meets the oxygen-rich water of the deep ocean, a chemical gradient is established, a difference in chemical potential between the sulfide-rich vent fluid and the oxidized surrounding water. This gradient is energy. It is the same kind of energy that a battery stores, a chemical potential difference that can be tapped to do work. And chemosynthetic microorganisms have evolved precisely to tap it. They catalyze the oxidation of hydrogen sulfide, the reaction between sulfide and oxygen, extracting the released energy to drive the synthesis of organic molecules. The efficiency of this process is remarkable. Chemosynthesis is not as energy-rich as photosynthesis, the energy available from sulfide. Oxidation is less than the energy available from sunlight, but it is sufficient to support the growth of dense microbial mats, sufficient, through the symbiotic relationships we described, to power the remarkable productivity of vent communities, sufficient to sustain life in one of the most physically extreme environments on Earth. The chemical landscape around a hydrothermal vent is complex and constantly shifting. Different chemical gradients exist at different distances from the vent. Hydrogen sulfide, concentration, temperature, oxygen levels, pH, and different microorganisms have evolved to exploit different positions within this gradient. Some prefer the hottest, most sulfide-rich environments, right at the vent opening. Others thrive in the cooler, more oxidized water further from the vent. Still others live at the interface between sulfide-rich and oxygen-rich water, precisely where the chemical gradient is steepest and the energy yield is greatest. This chemical zonation creates a spatial structure in the microbial community around the vent, a layering of different microbial types at different distances, each occupying the chemical niche to which its biochemistry is best suited. And within this microbial landscape, the larger animals make their livings, the tube worms and clams and mussels and crabs and fish, each adapted to a particular position and chemical environment, each part of a web of dependencies and interactions that makes the vent community a functioning ecosystem rather than a collection of independent organisms. There is something meditative about the chemistry of the deep. It does not hurry. It does not change from day to day or season to season. The chemical gradient around a vent is as steady as the vent itself, a constant, patient energy source that sustains its community with a reliability that the surface world, with its day-night cycles and seasonal fluctuations, does not always offer. Of all the organisms in the hydrothermal vent ecosystem, the tube worms are perhaps the most visually spectacular and the most scientifically remarkable. Riftia pachyptilla, the giant tube worm, is the species that first astonished the scientists aboard Alvin in 1977. It is an annelid worm, related distantly to the earthworms in garden soil, but it has evolved into something almost unrecognizable as a worm. Its white chitin tube, anchored to the rock around the vent, can reach 2 meters in length. From the top of this tube, a plume of bright red gill tissue extends into the water, an elaborate, feathery structure that looks almost like the petals of a strange flower and is rich in hemoglobin, the same oxygen-carrying molecule found in human blood, which gives it its striking red color. This plume is the tube worm's entire interface with the external chemical environment. It absorbs hydrogen sulfide, oxygen, and carbon dioxide from the water, all 3 chemicals needed by the chemosynthetic bacteria in the trophosome. The hemoglobin in the plume has evolved special binding properties that allow it to carry both oxygen and sulfide simultaneously, without the two reacting with each other. A chemical feat that required significant evolutionary refinement. The tube worm has no need to find food. It has no need to move. It sits in its tube, extending its plume into the vent current, and lets the bacteria inside it do the rest. In thriving vent communities, Riftia tube worms grow in dense clusters, thickets of white, tubes and red plumes that can cover large areas of the seafloor around the vent. These clusters create structure, physical habitat that other organisms colonize. Small crabs shelter in the spaces between tubes. Polychaete worms, small marine worms, weave through the colony. Shrimp graze on the microbial mats that grow on the tube surfaces. The tube worm colony is not just a community of tube worms. It is an ecosystem anchor, a habitat-forming structure that enables the existence of dozens of other species. The growth rate of riftia tubeworms is, for deep-sea organisms, remarkable. In the cold, resource-limited deep ocean, most organisms grow extremely slowly, but tubeworms at active vents, bathed in the chemical wealth of the vent fluid, can grow more than a meter per year, among the fastest growth rates of any marine invertebrate. This rapid growth allows them to colonize newly formed vent sites quickly, establishing the structural framework that other species will colonize in turn. The relationship between vent activity and tubeworm community is dynamic. A newly activated vent will be colonized first by microbial mats, then by mobile grazers, then by settling tubeworm larvae, then by the full suite of other species that depend on the structural habitat the tubeworms provide. As the vent ages and its output changes, the community composition shifts accordingly, and when the vent finally goes inactive, the tubeworm colony dies, the chemical gradient fades, and the seafloor returns to the quiet, sediment-draped state of the surrounding abyss. The tubeworm forests of the deep ocean floor are thus temporary, born with the vent activity that sustains them, dying when that activity ceases. But on the timescale of a human life, or even many human lifetimes, they appear permanent. The oldest known individual tubeworms are estimated to be hundreds of years old. The communities they anchor have existed at some vents for centuries. Beyond the tubeworms, the hydrothermal vent communities host a cast of creatures that are, almost without exception, extraordinary. The giant clams that first accompanied Alvin, scientists on their 1977 dive, belong to the genus Calyptogena. Like the tubeworms, they harbor chemosynthetic bacteria in their tissues, in their gills, in this case, and derive most of their nutrition from the organic compounds these bacteria produce. They can reach lengths of 25 to 30 centimeters, enormous by the standards of deep-sea bivalves, and they live in dense clusters, sometimes numbering thousands of individuals, arranged with their siphons directed toward the vent flow to maximize their exposure to the sulfide-rich water. The mussels of the deep vent communities, belonging to genera like Bathymodiolus, are similarly oversized compared to their shallow water relatives, also sustained by chemosynthetic bacteria in their tissues. They are more tolerant of variable chemical conditions than the clams and tend to colonize areas of diffuse vent flow, where the chemical concentrations are lower and more variable. The vent shrimp are perhaps the most visually striking of the mobile vent fauna. At vents along the mid-Atlantic ridge, swarms of tiny shrimp called Rimicaris exoculata, literally the eyeless vent shrimp, congregate in extraordinary density around the black smoker chimneys. They have no functional eyes in the conventional sense, the image-forming eyes that most crustaceans use to navigate in lighted environments would be useless in the absolute darkness of the vent. Instead, they have evolved a pair of light-sensitive organs on their backs, not eyes in the usual sense, but organs capable of detecting the faint glow of thermal radiation emitted by the superheated vent fluid. They use this thermal sense to navigate around the vent, staying close enough to the warm, sulfide-rich fluid to feed without getting so close that they are cooked by the extreme heat. What do they eat? The shrimp graze on the chemosynthetic bacteria that coat the surfaces of the vent chimneys, essentially farming the microbial mats that grow in the mineral-rich environment. They also harbor chemosynthetic bacteria in their gill chambers, a partial internal food supply in addition to their external grazing. The density of shrimp around some mid-Atlantic black smokers is extraordinary. Thousands of individuals packed so closely that they form a continuous living carpet over the chimney surface, jostling for position in the sulfide-rich currents. The vent crabs, pale, ghostly creatures that patrol the periphery of the vent communities, are more conventionally predatory. They feed on the other organisms of the community, scavenging dead and dying tubeworms and clams, preying on the smaller mobile fauna. They are the scavengers and hunters of the vent ecosystem, playing the role that crabs and other mobile predators play in shallow water marine communities. Vent fish tend to be small and cryptic, eelpouts and zoarchids that shelter in the crevices of the vent structures, feeding on the smaller organisms of the community. Some vent fish have independently evolved the ability to tolerate the warm, sulfide-rich conditions near the vents, a feat that requires physiological adaptations not yet fully understood. Octopuses occasionally visit vents, drawn, apparently, by the abundance of prey concentrated in the vent community. They are transient visitors rather than permanent residents, representing the connection between the vent ecosystem and the broader deep ocean. And throughout all of this, the microbial mats continue. The chemosynthetic bacteria, archaea coating every available surface in thin, layered films of microscopic life, the primary producers upon which everything else ultimately depends. Among the microorganisms of the hydrothermal vent, one group deserves particular attention. Archaea, the third domain of life, distinct from both bacteria and eukaryotes, are the masters of extreme environments. They were once thought to be simply an unusual group of bacteria, but molecular biology revealed in the 1970s and 1980s that archaea are as different from bacteria as bacteria are from animals. They are a completely distinct form of life, with their own evolutionary history, their own biochemical toolkit, and their own unique adaptations. Many archaea are extremophiles, organisms that not only tolerate but require extreme conditions. Thermophilic archaea thrive at temperatures far above the boiling point of water at surface pressure. Hyperthermophilic archaea have their optimal growth temperatures above 80 degrees Celsius, with some species growing best at 105 or even 121 degrees, the highest temperature at which any organism has been shown to grow and reproduce. At hydrothermal vents, these hyperthermophilic archaea are found in and around the hottest vent openings, environments that would instantly kill virtually any other known organism. They have evolved proteins and membrane lipids that remain stable and functional at temperatures that denature the proteins of organisms adapted to cooler conditions. One group of archaea found at hydrothermal vents, the methanogens, produce methane as a metabolic byproduct. They do not use hydrogen sulfide as their energy source, but instead combine hydrogen and carbon dioxide to produce methane and water. Their methane production represents another chemical cycle within the vent ecosystem, adding to the complexity of the chemical environment. Other vent archaea are involved in the nitrogen cycle, transforming nitrogen-containing compounds in ways that make nitrogen available to the rest of the community. Still others are involved in iron and sulfur cycling. The microbial community at a hydrothermal vent is not a simple collection of chemosynthetic sulfide oxidizers. It is a complex consortium of different metabolic types, each playing a different role in the cycling of elements through the ecosystem. The discovery of hyperthermophilic archaea at hydrothermal vents has had implications for our understanding of the origins of life, the deepest branches of the tree of life. The organisms most closely related to the common ancestor of all life on Earth are consistently thermophiles and hyperthermophiles associated with high-temperature environments. This pattern suggests that life may have originated in high-temperature hydrothermal environments, rather than in the warm ponds and shallow seas that earlier origin of life hypotheses favored. If life began at hydrothermal vents, at the chemical interface between hot mineral-rich fluid and cooler oxidized water, then the deep ocean ecosystems we are exploring tonight are not just the most ancient-looking ecosystems on Earth. They may be, in some deep sense, the most ancient, the successors in the same environments of the very first communities of living things on this planet. Hydrothermal vents are not the only places in the deep ocean where chemical energy sustains communities independent of sunlight. In the 1980s, scientists discovered another type of chemosynthetic deep-sea ecosystem, in some ways even more extensive and diverse than the vent communities. Cold seeps, also called cold vents or methane seeps, are places on the seafloor where fluids, rich in methane, hydrogen sulfide, and other reduced chemicals, seep slowly upward from sediments or from fissures in the rock. Unlike hydrothermal vents, these fluids are not hot. They emerge at temperatures close to the surrounding seafloor water. There is no dramatic chimney, no billowing black smoke. Instead, the seeping fluid rises quietly, invisibly, through the sediment, supporting a community of chemosynthetic microorganisms and the animals that depend on them. Cold seeps are found along the margins of continents, at the edges of the continental shelves, where thick accumulations of sediment rich in organic material have been buried and compressed over millions of years, producing methane and other hydrocarbons that slowly migrate upward to the seafloor. They are also found at other geological settings, subduction zones, where one tectonic plate slides beneath another, squeezing water and gases from the descending sediments. And they are found at mud volcanoes, places where sediment and fluid are pushed upward to the seafloor by geological pressure, forming structures that look, on a small scale, like the volcanic features of the surface world. The communities at cold seeps share many characteristics with hydrothermal vent communities. Chemosynthetic bacteria and archaea are the primary producers, supported by the chemical energy of the seeping fluids. Giant tube worms are present at some cold seeps, along with chemosymbiotic mussels and clams similar to their vent counterparts. Mobile fauna, crabs, shrimp, fish, octopuses, congregate around the chemical richness of the seep. But cold seep communities have some distinctive features that reflect their different chemical environment. One of the most remarkable is the carbonate rock formations that often develop around cold seeps. As archaea in the sediment around the seep consume methane, they produce bicarbonate as a byproduct. This bicarbonate reacts with calcium in the seawater to form calcium carbonate, limestone, which precipitates from the water and accumulates around the seep, building irregular formations of carbonate rock over thousands of years. These carbonate formations are important in several ways. They create physical habitat, hard surfaces where corals, sponges, and other sessile organisms can attach. They preserve the record of past seep activity, carbonate deposits that formed thousands or millions of years ago when a now inactive seep was flowing. And they are a significant carbon sink, one of the ways in which carbon from deeply buried, organic material is eventually sequestered in rock. Some cold seep communities are extraordinarily long-lived. Certain cold seep sites on the continental margins of the Gulf of Mexico and elsewhere appear to have been active for thousands of years, supporting communities that have had thousands of years to develop their ecological complexity. The tube worms at some Gulf of Mexico cold seeps are estimated to live for more than 200 years, and some individuals may be more than 300 years old, making them among the longest-lived animals known. The slow pace of cold seep ecosystems is one of their defining characteristics. Cold temperatures, reduced chemical gradients relative to hot vents, and the gradual, diffuse nature of the seeping fluid create conditions where life moves at a slower tempo than at the high-energy vents. Growth rates are lower, community development is more gradual, the ecological time frame is measured in centuries rather than decades. There is a quality of deep patience to the cold seep communities, a quality that resonates with the night we are spending together, drifting through these quiet, ancient, unhurried worlds. There is another type of deep-sea ecosystem that deserves a place in our journey tonight. One that has a particular quality of quiet grandeur, the whale fall. When a whale dies in the ocean, its body sinks to the seafloor. This is an event of enormous ecological significance. A large whale can weigh tens of thousands of kilograms, a concentration of organic material far greater than what arrives at any equivalent area of the deep seafloor through the normal rain of marine snow. A whale fall is, in effect, a sudden, dramatic delivery of biological wealth to a world that is ordinarily resource-limited. The ecological succession on a whale fall has been studied in detail, and it unfolds in stages that can last for decades. In the first stage, the mobile scavenger stage, large mobile organisms arrive to consume the soft tissue of the whale. Sleeper sharks, hagfish, rat-tailfish, and various invertebrates converge on the carcass, sometimes in large numbers, They can consume tens of kilograms of tissue per hour. The soft tissues of a large whale may be stripped to the bone within months. In the second stage, the enrichment opportunist stage, the exposed bones and the sediment around the carcass become colonized by dense populations of opportunistic organisms attracted by the nutrient-rich environment. Polychaete worms, crustaceans, and other invertebrates exploit the concentrated organic material. But the third stage is where the whale fall ecosystem becomes something truly remarkable. As the remaining soft tissue within the bones is consumed by bacteria, the lipid-rich whale bone itself becomes the substrate for chemosynthetic communities. Bacteria colonize the bone, breaking down the fats and releasing hydrogen sulfide as a metabolic byproduct. And where there is hydrogen sulfide, there is the potential for the same chemosynthetic ecosystem we have been exploring at hydrothermal vents. Specialized chemosymbiotic mussels and clams colonize whale bones. A species of tubeworm called Osedax, the bone-eating zombie worm, drills into the bone with root-like structures, hosting bacteria that break down the bone lipids. The whalebone becomes, in effect, a small-scale cold seep, a chemosynthetic island on the seafloor, sustained not by geological processes but by the accumulated biological wealth of a single large animal. This stage, called the sulfophilic or chemoautotrophic stage, can last for decades. Large whale skeletons may support chemosynthetic communities for 50 or even 100 years after the whale's death. Scientists have proposed that whale falls serve as stepping stones, islands of chemosynthetic habitat scattered across the vast abyssal plain that allow species to disperse between hydrothermal vents and cold seeps. Many species found at whale falls are related to species found at vents and seeps. The whale fall may be an intermediate habitat, a place where vent and seep organisms can survive and reproduce between the geological features that sustain longer-term chemosynthetic communities. The distribution of whale falls across the deep ocean floor depends, of course, on where whales go when they die. Before commercial whaling dramatically reduced whale populations in the past 19th and 20th centuries, whale falls may have been far more common. Scattered more densely across the seafloor, the stepping stones more closely spaced. The loss of whale populations through whaling may thus have had consequences for the deep sea that extend far beyond the obvious impact on whale communities themselves. This thought, that the reduction of whale populations affects the ecology of the deep ocean floor through the reduction of whale falls, is one of those ecological connections that, once seen, seems both obvious and astonishing. Everything is connected, in the ocean as on land. The surface and the deep are not separate worlds but deeply linked ones, exchanging material and species and energy in ways that science is still mapping. When most people think of coral reefs, they picture the sunlit, color-saturated ecosystems of shallow tropical seas, the Great Barrier Reef, the coral gardens of the Caribbean, the bright fish and diverse invertebrates of the warm water reefs. But coral reefs also exist in the deep ocean. Cold water coral reefs, sometimes called deep-sea coral reefs or cold water coral ecosystems, grow on seamounts and ridges and the edges of continental margins at depths of 200 to 3,000 meters or more, in water temperatures of 4 to 12 degrees Celsius, in complete darkness. These deep coral ecosystems do not have the brilliant colors of tropical reefs. They lack the symbiotic algae that gives shallow water corals their hues, but they are no less structurally complex or ecologically diverse. Deep water corals can form reefs hundreds of meters thick and tens of kilometers across, built up over thousands of years of slow growth by individual coral colonies. The dominant deep-water reef-building coral in the North Atlantic is Lophelia pertusa, colonial coral that grows at about 4 to 25 millimeters per year, building, branching white or pale pink structures that accumulate over centuries into reef frameworks of considerable size. Individual Lophelia colonies can live for several hundred years, and the reefs they build collectively represent thousands of years of accumulation. These deep coral reefs are extraordinary repositories of biodiversity. The complex three-dimensional structure of the coral framework provides habitat for hundreds of species of invertebrates, sponges, sea fans, brittle stars, feather stars, crustaceans, polycate worms, and many others. Fish shelter in the reef crevices, while commercially important species like rockfish and certain deep-water sharks use the reefs as breeding grounds and nurseries. Because deep corals do not depend on sunlight, they do not need warm, clear, shallow water. They grow wherever the water brings them enough food, in the form of the small particles and organisms that drift with the currents, and where the substrate allows them to attach. Seamounts, underwater mountains, the tops of which may still be hundreds of meters below the surface, are often important sites for deep coral development because they deflect and accelerate currents, bringing the deeper, particle-rich water upward and creating particularly food-rich conditions. The ancient nature of deep coral reefs makes them extraordinary scientific archives. The skeletons of corals record the chemical composition of the seawater in which they grew, the temperature, the acidity, the concentration of various elements, with a precision that allows scientists to reconstruct past ocean conditions from coral cores, much as ice cores allow reconstruction of past atmospheric conditions. Deep coral records extending thousands of years into the past have provided valuable data on how the ocean has changed over time. They are also among the most vulnerable marine ecosystems to human disturbance. Deep-sea bottom trawling, the practice of dragging heavy fishing nets along the seafloor, can devastate deep coral reefs, destroying in minutes reef structures that took centuries to build. Recovery, where it occurs at all, requires decades to centuries. The damage to many deep coral ecosystems from bottom trawling is well documented and represents one of the most significant impacts of human activity on the deep ocean. Let us drift away from the vents and reefs for a while and spend some time on the abyssal plain. The abyssal plain is the deep ocean floor away from the ridges and margins, the vast, flat expanses that cover much of the ocean bottom at depths of 3,000 to 6,000 meters. They are, by area, the most extensive ecosystems on Earth. They are also, superficially, some of the least eventful-looking places on the planet. The sediment of the abyssal plain is, in most places, extraordinarily fine. A soft, pale ooze, composed of the accumulated remains of countless microscopic organisms that lived in the surface waters above and drifted downward after their deaths. The most common component in many areas is the skeletal remains of foraminifera, tiny, single-celled organisms with intricate calcium carbonate shells that rain down from the surface in prodigious quantities. In other areas, the sediment is dominated by the silica shells of diatoms and radiolarians. This sediment accumulates at rates so slow that they are almost incomprehensible. A centimeter of sediment may represent thousands of years of deposition. The deepest layers of abyssal sediment cores are millions of years old, records of biological activity in surface waters that predates the existence of our species by enormous margins. Walking, if such a thing were possible, on the abyssal plain would be like walking on the softest, finest powder imaginable. Your feet would sink. The sediment would billow around you in slow, lazy clouds that would take a very long time to settle in the still, dense water of the deep. On this soft, pale surface, life proceeds at a pace appropriate to its setting. Sea cucumbers, holothurians, are among the most abundant animals on the abyssal plain. These elongated, soft-bodied relatives of sea stars and sea urchins move slowly across the sediment surface, hoovering up sediment with their feeding tentacles, processing it through their digestive systems for the organic material within, and depositing the cleaned sediment behind them in characteristic trails and furrows. Some abyssal plains are marked everywhere with the trails of sea cucumbers, a living calligraphy written in sediment, recording the slow passage of these ancient creatures across the floor. Brittle stars, ophiuroids, sprawl across the sediment, their long, flexible arms extending in all directions, capturing particles of organic material from the water immediately above the seafloor. They are more active than sea cucumbers, capable of moving at several centimeters per second when motivated, but in the absence of urgency they too move slowly, deliberately, in keeping with the tempo of the deep. Polychaete worms tunnel through the sediment, bioturbating it, mixing the layers, moving organic material from the surface downward, creating burrows that other organisms will later inhabit. This bioturbation is ecologically important, cycling nutrients through the sediment and maintaining the chemical conditions that allow organic material to be broken down efficiently. And everywhere, invisible to the naked eye but present in enormous numbers, the bacteria. The deep sediments contain bacterial populations of extraordinary density, billions of cells per cubic centimeter, that carry out the slow decomposition of organic material, cycling carbon and nitrogen and phosphorus through the abyssal ecosystem. The abyssal plane is not empty, it is not dead, it is simply slow, operating on timescales and at intensities that are difficult for human perception to appreciate. But it is alive, quietly and thoroughly, across the entire vast floor of the ocean. Let us return to the water column, to the open water of the deep ocean, away from the seafloor, and spend some time with the creatures that live there. The midwater, the vast volume of ocean water between the sunlit surface and the seafloor, is home to an extraordinary diversity of organisms, many of them unfamiliar and strange by the standards of the surface world. Bioluminescence is perhaps the most striking characteristic of midwater life. In the darkness of the deep ocean, where no sunlight penetrates, the production of light by living organisms is extraordinarily widespread. Estimates suggest that more than 75% of deep-sea organisms are capable of producing light, making bioluminescence the most common form of communication and interaction in the largest living space on Earth. The light is produced by a chemical reaction, the oxidation of a compound called luciferin by an enzyme called luciferase that releases energy as photons rather than heat. The reaction can occur in specialized light-producing organs called photophores, in bacteria living symbiotically within the organism, or in chemicals released into the water. Deep-sea bioluminescence serves many functions. Anglerfish use a bioluminescent lure, grown from a modified dorsal spine, illuminated by symbiotic bacteria, to attract prey in the dark. Counter-illumination, the production of light on the underside of an organism that matches the faint downwelling light from above, allows some midwater organisms to become effectively invisible to predators looking upward. Communication, mate attraction, predator startling, prey attraction. The diversity of functions for which deep-sea organisms have evolved bioluminescence is remarkable. The siphonophores, colonial organisms related to jellyfish, are among the most extraordinary inhabitants of the deep midwater. Each siphonophore is not a single organism, but a colony of hundreds or thousands of individual zooids, each specialized for a different function: feeding, reproduction, movement, defense. The colony is coordinated as a single unit, yet it is made of many genetically identical individuals. The boundary between individual and colonial organism blurred in a way that challenges ordinary biological categories. The longest known siphonophore, Praia dubia, can reach 40 meters in length, longer than any other animal. It drifts through the midwater in a long, ghostly chain, its feeding tentacles trailing outward to capture small crustaceans and fish. Ctenophores, comb jellies, fill the midwater with their transparent, iridescent bodies, beating rows of fused cilia that diffract light into rainbow patterns in the rare occasions when any light reaches them. They are voracious predators, consuming copepods and other small crustaceans in enormous quantities. The vampire squid, Vampiroteuthis infernalis, literally the vampire squid from hell, is a unique cephalopod that inhabits the oxygen minimum zone of the deep ocean, though between 600 and 900 meters, in waters so oxygen-depleted that most other active predators cannot survive there. It has the largest eyes relative to body size of any animal on Earth, enormous blue-green orbs that gather every available photon in the dim twilight of its environment. Despite its alarming scientific name, it is not a particularly aggressive predator. It feeds primarily on marine snow and small particles. using bioluminescent tips on its arms to create complex light displays. These are some of the inhabitants of the Midnight Zone, a world that is, in some ways, more alien than the surface of any other planet we know, and yet is here, on Earth, in the ocean that covers most of our world, accessible if we are willing to descend. Below the abyssal plain, in the deepest places on Earth, the ocean trenches, where one tectonic plate dives beneath another, lies the Hadal Zone, named for Hades, the Greek underworld. The Hadal Zone encompasses ocean depths below 6,000 meters. It is defined not just by depth but by geography. Hadal environments exist primarily in the deep, trenches formed at subduction zones, narrow, elongated features that plunge to extraordinary depths. The Mariana Trench is the deepest of these. At Challenger Deep, its deepest point, the ocean floor lies approximately 10,900 meters below the surface. The pressure there is approximately 1,100 times the pressure at sea level, the equivalent of 50 jumbo jets stacked on top of a single square meter. For a long time, it was assumed that the extreme conditions of the Hadal Zone, the crushing pressure, the total darkness, the near-freezing temperatures, would support little life. The Hadal Zone was thought to be, if not sterile, then very nearly so. This assumption was wrong. The Hadal Zone hosts specialized communities of organisms that are found nowhere else. Hadal amphipods, small crustaceans, swarm through the trench waters in enormous numbers, feeding on organic material that accumulates in the trench due to its topographic depression. Holothurians, adapted to extreme pressure, live on the trench floor. Snailfish, small, translucent fish, have been recorded at depths below 8,000 meters, deeper than any other fish species. These Hadal organisms have evolved adaptations to the extreme pressure. At very high pressures, proteins lose their normal shapes. Cell membranes become too rigid to function, and the molecular machinery of life breaks down. Hadal organisms counteract this through the accumulation of small organic molecules, particularly a compound called trimethylamine oxide, that stabilize proteins and membranes against pressure-induced deformation. The concentration of trimethylamine oxide in the tissues of deep-sea fish increases with depth in a remarkably linear fashion, as if the organism is continuously calibrating its chemical defenses to exactly match the pressure of the environment it inhabits. The deep trenches also concentrate organic material. The topographic depression of a trench acts as a funnel, collecting the sediment and organic particles that drift down from the surrounding abyssal plain. Floors can therefore be surprisingly rich in organic material, supporting higher densities of deposit-feeding organisms than the abyssal plain above. The Hadal Zone remains one of the least explored environments on Earth. Only a handful of expeditions have reached the very deepest points, and the technology for conducting sustained scientific research at these depths is still being developed. What has been observed there has consistently exceeded expectations. More life, more diversity, more ecological complexity than the extreme conditions would lead one to predict. The deep ocean is, in this way, consistently surprising. It resists the expectation of simplicity. It insists on being more complex, more alive, more interesting than our models predict. One of the qualities of the deep ocean that most profoundly distinguishes it from the surface world is its relationship with time. At the surface, time is structured by light, day and night, the rhythm of the tides, the seasons. These are the temporal frameworks within which surface life organizes itself, the clocks that tell organisms when to be active, when to rest, when to breed, when to migrate. In the deep ocean, these temporal cues are absent. There is no day or night below the photic zone. There are no seasons in the temperature or light regime, though there are seasonal pulses in the delivery of organic material from the surface above, reflecting the seasonal cycles of productivity in the surface ocean. The deep ocean is not entirely timeless, but its rhythms are muted, slower, less immediate than those of the surface world, and the organisms of the deep reflect this in their biology. Deep-sea organisms are, in general, extraordinarily long-lived, The giant tube worms of cold seeps live for centuries. Deep-sea sharks grow very slowly and may live for hundreds of years. The Greenland shark, which reaches depths of more than 2,000 meters, has been estimated to live for more than 400 years, making it the longest-lived vertebrate known. The deep-sea clam Arctica islandica, the ocean quahog, can live for more than 500 years. Black coral colonies in the deep ocean can live for thousands of years. These extraordinary lifespans reflect the combination of cold temperatures, which slow metabolism and reduce the rate of cellular damage, and the absence of the dramatic environmental fluctuations that in shallower environments cause periodic die-offs and require rapid life cycles to ensure survival. Growth in the deep ocean is correspondingly slow. A deep-water coral that adds a centimeter of growth in a year is doing well. A fish that takes 20 years to reach reproductive maturity is not unusual in the deep sea. The pace of biological process is calibrated to the pace of the environment, unhurried, patient, extended over timescales that human lives rarely permit us to witness. This slowness has consequences for the resilience of deep-sea ecosystems to disturbance. Because organisms are long-lived and slow-growing, populations recover slowly from damage. A deep-sea coral reef destroyed by bottom trawling will not recover in human lifetimes. A population of deep-sea sharks reduced by targeted fishing may take centuries to rebuild. The patience of the deep ocean is, in this sense, also a vulnerability. It cannot recover at the pace that human exploitation currently imposes. But in the absence of such disturbance, the deep ocean's relationship with time is one of its most profound qualities. These communities persist, essentially unchanged in character, for longer than human civilizations. The communities of tube worms and clams and chemosynthetic bacteria around a stable cold seep have been cycling through the same ecological processes, the same succession of birth and growth and death and renewal, for longer than any human institution has existed. There is a depth of time here that is humbling to contemplate. And in the context of tonight's journey towards sleep, perhaps there is something in it to draw comfort from. The world goes on. The deep ocean proceeds. Time, in its deepest dimensions, is vast and accommodating, and not in any particular hurry. The discoveries made in the deep ocean over the past half-century have fundamentally changed the definition of the habitable. Before the discovery of hydrothermal vent ecosystems in 1977, the limits of life were thought to be relatively well understood. Life required sunlight, directly or indirectly, as its energy source. Life required liquid water. Life required temperatures within a fairly narrow range. Life required modest pressures. Life required certain chemical conditions: neutral or mildly acidic pH, reasonable oxygen levels, absence of toxic compounds. The deep ocean has violated every one of these assumptions. Life exists at hydrothermal vents without sunlight. Life exists in the crushing pressures of the Hadal zone. Life exists at temperatures above 100 degrees Celsius at vent openings. Life exists in highly acidic vent fluids. Life exists in the presence of hydrogen sulfide concentrations that would be rapidly lethal to surface organisms. Life exists in the near-total absence of oxygen in the oxygen minimum zones of the ocean. Each of these discoveries has expanded the envelope of the habitable. Each has extended the range of conditions under which we know life can exist. Each has made the universe more habitable, in principle, than we previously thought. And the implications extend beyond Earth. The icy moons of Jupiter and Saturn, Europa, Enceladus, Ganymede, and possibly others, are believed to harbor liquid water oceans beneath their icy surfaces. These oceans are cold, dark, and pressurized. They may have hydrothermal systems at their seafloors. They may have chemical gradients between reduced and oxidized regions. These are precisely the conditions under which life exists in Earth's deep ocean. The discovery that chemosynthetic life can thrive in the conditions of the deep ocean has transformed the assessment of the possibility of life on icy moons from a speculative notion to a scientifically serious proposition. If life can arise and sustain itself without sunlight, using chemical energy, in cold, high-pressure, dark environments, then the dark ocean beneath Europa's ice becomes not just a possible habitat for life, but a plausible one. We cannot yet know whether life exists in those distant oceans, but we know, now, that if it does, there is no physical or chemical reason why it could not. The conditions that seem hostile are the same conditions under which life thrives in our own planet's deep. This is perhaps the deepest scientific legacy of the exploration of the deep ocean floor. Not any specific discovery about any specific organism or ecosystem, but the expansion of the imaginable. The demonstration that life is more robust, more versatile, more inventive than we knew, that the universe contains more places where life might exist than we previously dared to suppose. The deep ocean communicates in ways that are very different from the surface world. At the surface, the primary sensory currency of most animals is light. Vision, the detection and interpretation of electromagnetic radiation in the visible spectrum, dominates the sensory lives of most surface animals, including ourselves. The world of the sunlit ocean is a world of color and form, of visual signals and visual cues. In the deep ocean, light is scarce or absent, and the sensory world is organized around other modalities. Sound travels extraordinarily well in water, faster than in air, and over much greater distances. The deep ocean has a layer called the SOFAR channel, sound fixing and ranging channel, where the Ryan physical properties of the water create conditions that allow low-frequency sound to travel for thousands of kilometers with relatively little loss of energy. Blue whales calling in one part of the ocean can potentially be heard by other blue whales thousands of kilometers away. The deep ocean is not silent. It is filled with sound. The low, complex songs of whales, the crackling of shrimp, the distant roar of submarine earthquakes, the grinding of tectonic plates. These sounds travel through the water at the speed of sound, about 1,500 meters per second, reaching across distances that no light signal could traverse. Deep-sea organisms have evolved diverse sensory systems attuned to the information available in their environments. The lateral line system, present in fish and some other aquatic organisms, detects pressure, waves and water movement, allowing organisms to sense the presence of other animals or obstacles without needing to see them. Many deep-sea organisms have exceptionally sensitive chemical senses, able to detect trace concentrations of chemicals in the water that signal the presence of food, predators, or mates. And then there is bioluminescence, not just as a source of light for communication, predation, but as a sensory challenge. In the deep ocean, any emitted light is immediately conspicuous. A bioluminescent organism advertising itself for communication takes a risk. It also advertises its presence to predators. The evolution of bioluminescence in the deep ocean has been shaped by this tension between the benefits of communication and the risks of visibility. The result is a sophisticated ecology of light in the dark. Organisms that can produce light, detect it, and interpret it, using a medium that exists only because they themselves create it, in a world that is otherwise entirely dark. The deep ocean is one of the great repositories of Earth's history. The sediments of the deep seafloor contain a record of conditions on the Earth's surface extending back millions of years, a record preserved in the chemical composition of shells, in the species of organisms that lived and died in the surface waters above, in the isotopic composition of organic and inorganic material. Deep-sea sediment cores, cylinders of sediment extracted from the ocean floor by research. Ships, sometimes extending many meters deep and representing millions of years of accumulation, are among the most important archives of past climate and environmental conditions available to science. They record the glacial cycles of the ice ages, the mass extinction events that have punctuated the history of life, the long-term changes in ocean chemistry and circulation that have shaped the history of life on Earth. The chemistry of the deep ocean itself, the temperature, the dissolved oxygen content, the acidity, the circulation patterns, has changed dramatically over geological time, and those changes are recorded in the sediments. The deep ocean has been warmer and more sluggish in the past, During times when there was no ice at the poles to drive the cold, dense water formation that currently fuels the global ocean circulation. It has been colder and more vigorously circulating during ice ages. It has been more acidic during periods of high atmospheric carbon dioxide, and less acidic during periods of lower carbon dioxide. These changes have had profound consequences for the deep-sea communities we have been discussing. Shifts in ocean circulation affect the delivery of organic material to the deep from the surface. Changes in ocean chemistry affect the chemistry of vent fluids. Shifts in the position of mid-ocean ridges and subduction zones alter the distribution of hydrothermal vents and cold seeps. The communities of the deep ocean are not static, they change with the Earth over geological time, and right now, the deep ocean is changing again. Human activities are warming the planet, acidifying the oceans, altering ocean circulation, and reducing oxygen levels in some regions. These changes are reaching the deep ocean more slowly than they are affecting the surface, but reaching it nonetheless. The deep ocean is responding to human civilization, even though its residents have never seen a human, never heard a human voice, never experienced anything of human activity directly. The connection is real, even if it is indirect. The CO2 that enters the atmosphere from the burning of fossil fuels dissolves in the ocean and changes its chemistry. The warming of the surface ocean affects the formation of the cold, dense water that drives deep circulation. The changes ripple downward, slowly, over years and decades and centuries. The deep ocean is not apart from our world. It is part of our world, connected to everything we do on the surface, affected by choices made by billions of people who have never given the deep a second thought. This is something worth knowing. Even on a quiet night like this one, the world is more connected than it appears. We have spent much of tonight in darkness. The darkness of the deep ocean is not the darkness of a room with the lights off, which is relieved by the faint ambient light that finds its way around curtains and under doors. Not the darkness of a moonless night, which is pierced by stars. The darkness of the deep ocean, below the photic zone, away from any bioluminescent organism, is absolute. It is the absence of every photon, a darkness so complete that it has persisted in many parts of the deep ocean since those waters were last exposed to light millions of years ago. And in that darkness, life has found a way. This is one of the most extraordinary facts about the deep ocean. The darkness that we might instinctively associate with absence, with emptiness, with the inhospitable, is not those things. It is full, it is abundant, it is ancient and complex, and thriving in its own quiet way. The organisms of the deep have not missed the light. They evolved in the dark. The dark is their home. The dark is what they are built for. The pressure that would crush an unprotected human body is simply the ambient pressure of their lives. The cold that would be fatal to a surface organism is simply the temperature of existence. This adaptability of life, this capacity to find a home in conditions that seem to exclude life, is one of the most profound messages that the deep ocean sends upward to the surface world. We think of life as fragile, and in some ways it is. Individual organisms are easily killed. Individual species can go extinct. Individual ecosystems can be disrupted and degraded. But life as a phenomenon, the pattern of chemistry organizing itself into self-replicating, energy-harvesting structures, is extraordinarily robust. It has persisted for nearly 4 billion years on this planet, through mass extinctions and ice, ages, and dramatic shifts in atmospheric chemistry and ocean chemistry. It has colonized every environment that offers liquid water and a source of energy. It has found a way. The deep ocean reminds us of this. The communities around the hydrothermal vents, the long-lived corals and clams of the cold seeps, the strange, bioluminescent inhabitants of the midnight zone, all of these are expressions of life's fundamental tenacity, life's willingness to go to the dark places and find them habitable. And in the dark of tonight, the ordinary human dark of a room at night, the gentle dark that is simply the turning of the earth away from the sun, perhaps there is something of the same quality available. The dark is not empty. The quiet is not absence. The stillness of sleep is not a withdrawal from life, but a deepening of it. The body continuing its work in the dark, the mind releasing what it needs to release, and consolidating what it needs to keep. The deep ocean sleeps in a manner of speaking. Its slow processes, its long-lived communities, its patient cycling of elements through sediment and water. This is a kind of sleep, a long, deep, productive rest. You are part of the same living world. The deep ocean remains, in most of its extent, unknown to us. We have maps of perhaps a fifth of the deep seafloor at sufficient resolution to reveal its major features. We have visited, with submersibles and remote vehicles, a tiny fraction of the deep. The organisms we know are almost certainly a small minority of those that exist. The ecosystems we have described, the hydrothermal vents, the cold seeps, the deep coral reefs, are the ones we happen to find in the places we happen to look. There are almost certainly other types of deep-sea ecosystems waiting to be discovered. The tools for deep-sea exploration are improving. Autonomous underwater vehicles, AUVs, can now be programmed to survey large areas of the seafloor without the continuous attention of a surface ship. Remotely operated vehicles, ROVs, equipped with high-definition cameras and sophisticated sampling equipment, can conduct detailed investigations of specific sites. New sensor technologies allow the detection of chemical signals, biological markers, and physical properties that reveal the presence of interesting features before a vehicle is even deployed. The next decades will likely see a significant expansion of our knowledge of the deep ocean, in the breadth of areas surveyed, in the depth of investigation at known sites, and in the discovery of new types of environments and communities that we have not yet imagined. This exploration is not merely scientific curiosity, though it is certainly that. The deep ocean is connected to the climate system, to the global cycles of carbon and nitrogen and phosphorus, to the productivity of fisheries and the health of the ocean as a whole. Understanding it is part of understanding the Earth system, the complex, interconnected system of ocean and atmosphere and biosphere that sustains the conditions for life on the tree surface. And it is, simply, one of the last great unknown territories on this planet. There are still places that no human eye has seen, no instrument has visited, no mind has comprehended. That is a privilege, to live in a time when such places still exist, when such discoveries are still possible. We have traveled far tonight, from the warm, sunlit surface of the ocean, down through the twilight zone and the midnight zone, to the seafloor and the extraordinary communities that gather there. We have visited the hydrothermal vents with their black smokers and tubeworm forests. We have drifted over cold seeps and their ancient clam communities. We have paused at the whale falls, those temporary islands of biological wealth on the abyssal plain. We have looked into the darkness of the Hadal zone and found life there too. We have met organisms that live for centuries, communities that persist for millennia, ecosystems that have been cycling through the same processes since before our species existed. And we have encountered, again and again, the same fundamental theme: life's extraordinary capacity to find a home in places that seem inhospitable, to extract energy from sources that seem insufficient, to organize and persist and thrive in conditions that our surface-dwelling intuitions tell us should preclude it. The deep ocean is patient in a way that puts human impatience in perspective. The sea cucumber moving across the abyssal plain, processing its cubic centimeters of sediment per day, has been doing so. Its ancestors have been doing so for hundreds of millions of years. The chemosynthetic bacteria, oxidizing hydrogen sulfide at the base of a hydrothermal vent, are performing a reaction that their ancestors have performed since before the first eukaryotic cell existed. These are ancient processes, ancient communities, ancient forms of life, persisting in the dark at the bottom of the sea, patient and unhurried, while the surface world goes through its revolutions and extinctions, and the coming and going of civilizations. There is something deeply restful in that perspective, the long view of time, the quiet certainty of biological process continuing through the dark. These are the qualities of the deep ocean that, perhaps, make it the right companion for a night of rest. Somewhere below us, right now, the deep ocean continues its work. In the darkness beneath miles of water, tube worms extend their red plumes into the sulfide-rich currents of hydrothermal vents. Chemosynthetic bacteria oxidize hydrogen sulfide, slowly and steadily, as they have done since before our planet had oxygen in its atmosphere. Ancient clams filter the cold seep water through their gill tissues, harvesting the organic compounds produced by their symbiotic bacteria. Sea cucumbers move imperceptibly slowly across sediment that took millions of years to accumulate. None of this requires sunlight. None of it requires the turning of the earth, the warmth of the day, the activity of the surface world. It simply continues, deep and dark and patient, as it has continued for hundreds of millions of years. You too are about to continue something that does not require the activity of the day. Your body, in the coming hours, will perform the deep work of sleep, consolidating memories, repairing tissues, regulating hormones, clearing the metabolic byproducts of consciousness from your brain. This work does not need your attention or your effort. It simply happens, in the dark, while you are otherwise engaged. The deep ocean does not worry about its processes. The tube worm does not concern itself with whether the bacteria in its trophosome are performing their chemistry correctly. The cold seep community does not question whether the methane will keep flowing. These processes are trusted to proceed, and they do. You can trust your own deep processes tonight. Let your breathing slow to the rhythm of something ancient and patient. Let your heart find its own steady pace. Let the thoughts that circled through the day grow quieter, become less distinct. Sink gently, away like particles settling through kilometers of water toward the soft sediment of the salt, abyssal plain. The surface world will still be there when you wake. Your questions will still be there. Your responsibilities will still be there. But right now, in this dark and quiet hour, none of that is required of you. The only thing required is this: to rest, to sink, to let the dark be what the dark always is. Not an absence, but a presence. Not an end, but a continuation. Not a place where life stops, but a place where it deepens and slows, and does its most patient, most essential work. The ancient forests of the deep ocean floor are growing, slowly, in the dark below. And you are resting, gently, in the dark above. Both are part of the same living world. Both are exactly as they should be. Good night. Sleep deeply. The deep is patient, and so is the night, and so, tonight, are you.